This recording is the urinary system, part 2 of 2. This particular slide shows us the amount of substances filtered and reabsorbed by the kidneys in a 24-hour period. The first line that talks about water is measured in liters. Everything else is measured in grams. So with water, we should filter approximately 180 liters per day. That 108 is a typo and I could not fix it. And then the amount of water that's reabsorbed is going to be approximately 179 liters per day with a net output of 1 liter per day approximately. And of course that's all dependent upon intake. You'll notice that we do filter somewhere in the neighborhood of 10 to 20 grams of proteins, and all of that should be reabsorbed. If there is protein in the urine, that could indicate potential kidney damage. We will have an approximate output or amount of 3 grams of sodium in our urine over a 24-hour period and a 0.3 gram output of bicarbonate. There will be a little bit of glucose in the urine, 4 grams over a 24-hour period, as well as urea, potassium, and uric acid and creatinine. After filtrate is produced by blood flowing through the capillary tuft inside the glomerular capsule or Bowman's capsule, we have a filtrate, which is potential urine. And the filtrate that enters into the proximal convoluted tubule, that's what PCT stands for at this point, that proximal convoluted tubule is going to be all the filtrate, and that filtrate does not have formed elements, which are the red blood cells, white blood cells, and platelets, because those formed elements are too large to be filtered through the fenestrated capillaries in the glomerulus unless there's damage to those capillaries where big stuff is going through. Then that's a problem. But normally the filtrate is going to be everything that went through the capillaries minus the formed elements, which are cells, right, and platelets, and large proteins. Large proteins should not be filtered through, and they should not be going into the proximal convoluted tubule in the filtrate. So the proximal convoluted tubule is going to be the first point of modification of urine, because the simple cuboidal epithelial cells that line the proximal convoluted tubule have lots of microvilli on their surface for reabsorption and secretion. And so we're going to see that as this is the first point of urine modification, we're going to reabsorb all the substances that were initially absorbed by digestion. And as filtrate continues through the proximal convoluted tubule and then goes into the loop of Henle, at the proximal convoluted tubule, we're going to reabsorb things like sodium and chloride and calcium and amino acids and glucose and phosphate molecules. It's at this point where toxic substances and drugs would be secreted by the proximal convoluted tubule. So the things that we want to keep in are going to be reabsorbed. And again, that includes things like sodium chloride, calcium, amino acids, glucose, phosphate, magnesium. And as these things are reabsorbed, we're going to see that hydrogen ions are going to be antiported with the reabsorption of these other molecules. So you can see lots of hydrogen is going to be secreted as the other substances are reabsorbed. And our kidneys can get rid of hydrogen ions one at a time depending on concentration gradients. And this is how we can manage our pH very, very specifically. That's why drinking high pH water is kind of silly, because our bodies are so adept at managing acid-base balance with the kidneys, with the lungs, with chemicals in our bloodstream. So the high pH water is really kind of silly when you think about it, because it's kind of a moot point. So we have peritubular capillaries that are going to wrap around the proximal convoluted tubule so that substances that are reabsorbed can diffuse into the bloodstream, and the peritubular capillaries are going to continue as the proximal convoluted tubule continues to form the loop of Henle. Those peritubular capillaries are going to turn into vasorecta capillaries, and a lot of these reabsorbed substances are going to continue to move into our blood circulation. So remember that the capillaries within the vasorecta are going to operate under continuous high pressure that's coming from the afferent arteriole, which that lumen from the afferent arteriole can also be regulated in terms of its opening because of the juxtaglomerular cells, which are smooth muscle cells. They can constrict or relax to manage the pressure that's entering into the glomerulus. And then the peritubular and vasorecta capillaries are going to rely on osmolarity and concentration gradient of the substances that are moving in to the lumen of the proximal convoluted tubule. We've got all these substances which have a higher concentration gradient that's going to help them move back into the bloodstream. Now, sodium is actively pumped out of the proximal convoluted tubule and diffuses into the peritubular capillaries. And when sodium moves in from the potential urine back into the bloodstream, water will follow because water follows solute. It's an obligatory water reabsorption because water follows solute by way of osmosis. But please pay attention to the fact that in the peritubular capillaries we also have sodium, calcium, amino acids, glucose, phosphate, magnesium. All these other solutes are going to be reabsorbed in the proximal convoluted tubule, and they are going to often hitch a ride with sodium. That's co-transport. So we talk about co-transport of glucose, which is something that they like to talk about in nursing. Okay, so glucose and sodium are co-transported. But it's not the only molecule that's co-transported. Amino acids are co-transported with sodium. Phosphates co-transported with sodium. Magnesium comes in on its own, but calcium also gets co-transported with sodium, as does chloride. So a lot of things get co-transported with sodium, and then water likes to follow with all of these solutes and has that obligatory osmotic effect. I made a separate and distinct slide for glucose-sodium co-transport, which, by the way, symport means things are moving in the same direction. Antiport means that they're moving in the opposite direction. So symport and co-transport are sometimes used interchangeably because they're going in the same direction. I made a distinct slide because I was talking to some nursing students who had a problem with this concept, and I really want to make sure that anybody who's going into nursing understands that glucose and sodium are going to be co-transported. The glucose is basically taking a ride with the sodium molecule. So remember that almost all glucose, amino acids, vitamins are going to be recovered in the proximal convoluted tubule. And if circulating glucose levels exceed glucose transport capacity, because glucose has to be brought in with sodium, if they exceed the ability for the glucose molecules to be co-transported, in the proximal convoluted tubule, that's when glucose will appear in the urine, and that usually indicates diabetes because we don't want to see too much glucose in the urine. So again, glucose and sodium are going to be symported or co-transported. Sodium is going to move down its concentration gradient into the cell, taking glucose with it. Glucose will then enter the interstitial space by way of facilitated diffusion, and then it will continue on into the bloodstream. And the sodium-potassium pump is important because it's going to create that concentration gradient and help for sodium to continue to move into the bloodstream where water will follow. Bicarbonate molecules are super important when it comes to acid-base balance. In fact, recovering bicarbonate ions is key to balancing our levels of pH throughout our body. And this is because bicarbonate can undergo very fast reactions due to an enzyme known as carbonic anhydrase enzyme. And this is the same mechanism that works in erythrocytes with the co-transport of carbon dioxide, as well as in the stomach with the production of hydrochloric acid, and as well as within the pancreas in terms of its production of bicarbonate. So if we look at what's happening in the lumen of the proximal convoluted tubule, we can see that sodium is being actively transported into the proximal convoluted tubule cell, which is simple cuboidal epithelium, with the help of adenosine triphosphate. And when sodium is actively transported in, hydrogen ions are transported in antiport. Now, hydrogen ions will very quickly react with bicarbonate ions. When hydrogen and bicarbonate join together, they form a molecule known as carbonic acid. And carbonic acid is going to quickly be broken down into water and carbon dioxide. Now, please understand that carbonic anhydrase enzyme can break down carbonic acid. It can also form carbonic acid from the bicarbonate and hydrogen ions. It goes both ways. It goes in accordance with the gradient that's being generated by the hydrogen ions. Now, when the carbonic anhydrase enzyme splits carbonic acid into water and carbon dioxide, the water will continue on its journey to become potential urine. The carbon dioxide will diffuse into that proximal convoluted tubule cell and react with the water inside the cell with the help of carbonic anhydrase enzyme, as this reaction is now being driven by the carbon dioxide to form carbonic acid. And then again, the carbonic acid will be further broken down by the carbonic anhydrase enzyme into free hydrogen ions and bicarbonate. Bicarbonate will join with sodium that was reabsorbed here, and the sodium bicarbonate will be actively transported in antiport with hydrogen. And so the sodium bicarbonate will go into the bloodstream and the hydrogen will be pumped out of the bloodstream actively in antiport, and everything just keeps getting recycled and is driven by what's happening with the hydrogen and the bicarbonate so that everything can be balanced in terms of our pH. Our body balances our levels of pH so precisely that it's literally ion by ion that everything is being recycled and recovered. And note that in this, There's a sodium-potassium pump that is helping to create a sodium gradient, and there's solute recovery in the proximal convoluted tubule that helps promote the recovery of water also, so that not all of the water is lost. Remember, we reabsorb most of the water that gets filtered. After the potential urine has gone through the proximal convoluted tubule, its next journey is through a structure known as the loop of Henle. And parts of the loop are specialized to recover sodium and water that have been filtered by the glomerulus and gone through the proximal convoluted tubule. And within the loop, osmolarity, which is the concentration of solute in a solution, is going to change. The loop consists of two main portions. There is a descending limb or descending loop and an ascending limb or ascending loop. And you'll notice that the osmolarity in the beginning of this journey is higher as it starts in the descending loop than it is at the end in the ascending loop. So what happens is, as urine travels down the descending loop, water will leave that structure because there are permanent protein channels called aquaporins that are found along the membrane that make up that structure. So we've got permanent aquaporins, and that means that the descending limb is freely permeable to water. And you'll notice that the osmolarity actually increases as we go down that descending limb because we're losing water, but not solute. It's only permeable to water. So that means that the ratio of solute is increasing as water goes out and goes into the vasa recta. The vasa recta are straight capillaries. That's where they get their name. Vasa means vessel, rect means straight. So they're straight capillaries that are going to be found next to the descending limb of the loop of Henle. Another thing that I want to point out here is that the flow of urine is going in the opposite direction to the flow of the blood in the vasa recta. That forms what's referred to as a counter current. Counter means against. So a counter current mechanism means that the current or the flow of fluid in the vessels is moving in opposite directions. So in the descending limb of the loop of Henle, we go from an osmolarity concentration of 300 milliosmoles to 1200 milliosmoles as water is lost from the potential urine and goes into the vasa recta. You'll also notice at the bottom of the vasa recta has a higher concentration of solute that decreases when we get to the end of the vasa recta. Okay, so we're losing water from the descending limb of the loop of Henle to the vasa recta, so it's going into the bloodstream. So that's how we get water from the urine back into the bloodstream. Remember, we filter 180 liters per day, but we're reabsorbing 179 of that approximately. So the descending loop is freely permeable to water, impermeable to sodium chloride. However, urea can actually be excreted from the bloodstream into the descending limb of the loop of Henle. And that urea will actually be excreted by the collecting ducts. We'll take a look at that later. So again, the osmolarity in the descending limb is going to change as water is reabsorbed. Now, some solutes can go with the water, but it is impermeable to salt. Not a lot of solutes are going to go with it. Let's take a look at the next part of the loop of Henle. So now we've got the ascending portion of the loop, which is the ascending limb or ascending loop. And the ascending limb of the loop of Henle or ascending portion is completely impermeable to water now, but it is freely permeable to salt. And so that means that salt is going to go out, and that's going to decrease the osmolarity of the urine. Remember, we started at 300 milliosmoles as the urine began its journey through the loop of Henle. It became much more concentrated as we lost water, but now that we're losing salt, that osmolarity in the ascending portion really decreases. Now, salt is entering into the bloodstream, it's going into the vasa recta, so now that osmolarity in the vasa recta is going to start to increase as salt goes into the bloodstream. However, water is going to be pulled back in because water follows solute, and as water follows solute, it's going to be drawn to this high solute concentration that's been created in the vasa recta. And so water's going to follow that solute from the descending limb, where it's freely permeable to water. It's going to go back into the vasa recta. And what's kind of cool is that the osmolarity of the vasa recta ends up staying the same if we look at the beginning of the vasa recta to the end. Even though it changes throughout its journey, it ends up being equal, which is important. And we know that salt, which is sodium chloride, and not just salt but potassium, is going to go back into the vasa recta, and it can also go into the interstitium surrounding that loop of Henle. So we have lots of sodium-potassium pumps that lead to a hypotonic filtrate, which means the filtrate, the potential urine, is going to contain fewer solutes and become hypotonic. And that also creates hyperosmolarity in the medullary portion of the kidney, which is going to help us concentrate our urine. Because if we have more solute in the medullary portion of the kidney, that's also going to help concentrate the urine to a certain degree, So again, that's our countercurrent mechanism, by water following solute, by the flow of urine going in the opposite direction of the fluid in the blood vessels. And chlorine follows sodium because of leaky tight junctions in the ascending limb of the loop of Henle, and potassium can re-enter the lumen through leakage channels. However, the interstitial fluid and that's the fluid surrounding all these tubules and blood vessels, is going to become negatively charged, and that actually attracts the sodium, the potassium, calcium, and magnesium into the interstitial space, and that will ultimately attract those ions to move into the vasa recta. And so that's how more things will go back into our bloodstream. It's a lot going on. So there are thin and thick segments of the descending limb and ascending limb, and something to pay attention to is that the thick segments can move ions into the interstitial spaces for reabsorption. Again, I'll say that again, the thick segments can move ions into the interstitial spaces for reabsorption. Now, as I said before, the flow of filtrate in the loop of Henle is moving in one direction. It's moving in this direction, whereas the flow of blood is moving in the opposite direction, and that's what's called the counter current multiplier system. And so what happens is the descending limbs and ascending limbs, together with the capillaries, the vasa recta, side by side, have that fluid going in opposite directions. That's a counter current against the flow, opposite directions. Solute pumps are going to increase, multiply the concentrations of urea and sodium into the medullary portion of the kidney. And that helps slow down the movement of the flow so that the erythrocytes can gain and lose water without being damaged, so they don't get too much water and swell and lyse, nor do they lose too much water at the same time and crenate and shrink. So the movement of sodium and urea into the interstitial spaces by these mechanisms can create a hyperosmotic environment in the medullary portion of the kidney, and the net result of this countercurrent multiplier system is going to help recover water and sodium back into our bloodstream, back into circulation. Remember that peritubular capillaries are found around all of these structures, the proximal convoluted tubules, the loop of Henle, and the distal convoluted tubules, as well as in the medullary portion of the kidneys where we have the collecting ducts. And by the time the potential urine has reached the distal convoluted tubule, approximately 80% of the water that was initially filtered into Bowman's capsule has now been recovered by the time it reaches the distal convoluted tubule. And sodium can still be reabsorbed, in which case water will follow. And the distal convoluted tubule cells can also recover calcium in the presence of parathyroid hormone. When parathyroid hormone receptors bind to the cells in the distal convoluted tubule, that will cause calcium to be reabsorbed, and it will also stimulate calcidiol to be converted into calcitriol, which is the active form of vitamin D, because the parathyroid hormone will facilitate an enzyme called alpha-1 hydroxylase, and that's what converts calcidiol into calcitriol. We can also see that excess potassium and hydrogen ions, as well as urea, can be secreted into the potential urine in the distal convoluted tubules, and we know that urea can also be secreted into the collecting ducts to be excreted eventually. So urea can be secreted into distal convoluted tubules and collecting ducts, where it can be excreted with the urine. The collecting duct's main function is for water recovery. However, other things can happen in the collecting ducts. There are different kinds of cells in the collecting ducts, including principal cells, which have channels for the recovery or loss of sodium and potassium. There are also intercalated cells, which are designed to secrete or absorb acid or bicarbonate. And the collecting ducts, again, are basically going to regulate urine volume and osmolarity. They are regulated by antidiuretic hormone, which will insert aquaporins into those principal cells, pulling water from the collecting ducts, moving it back into the capillaries and into our bloodstream. Aldosterone can also work on the collecting ducts because it will cause sodium and potassium channels and pumps for sodium to become activated so that sodium moves out of the potential urine and back into the interstitium of the kidneys to be reabsorbed into blood vessels. And if plasma osmolarity rises, more water needs to be pulled from potential urine so it can go back into the bloodstream. If plasma osmolarity decreases, then less water is recovered and urine volume increases. So again, making sure that our plasma osmolarity is where it needs to be for water to move freely through blood vessels is really important. When it comes to regulating the flow of blood through the kidneys, there are two main mechanisms that do this. One is through the autonomic nervous system, and the other one is what we refer to as autoregulation. With the autonomic nervous system, we do not have dual innervation. We don't have sympathetic and parasympathetic innervation for blood vessels. We only have sympathetic stimulation. So the sympathetic nerves include the celiac plexus and the splanchnic nerves, which in the event that there's no sympathetic response, then blood vessels are going to be vasodilated. They're going to be at their normal opening, which is a state of vasodilation. In the event of a sympathetic response, that would cause vasoconstriction. That decreases the flow of blood through the glomerulus, which would trigger the renin, angiotensin, aldosterone system to come into play, where the collecting ducts are going to increase the reabsorption of sodium, which increases the reabsorption of water, which raises blood volume, increasing blood pressure. So not only do we have vasoconstriction throughout the entire body with sympathetic nervous system responses, with the exception of skeletal muscle because we need to be able to run away, but in terms of other organs, we're going to have vasoconstriction. It also is going to affect the kidneys. Now, in terms of autoregulation, there are two different mechanisms for autoregulation. Remember that arterioles have smooth muscle around them, and so when there is a stretch in that smooth muscle, there's going to be a reflexive contraction. And so that reflexive contraction with a little bit of stretch is going to kind of regulate the flow of blood, but it actually results in very little change in terms of overall flow. It's just a very local kind of stretch contraction mechanism. In terms of tubuloglomerular feedback, which is the main type of regulating blood flow for autoregulation, remember we have the juxtaglomerular apparatus, and sometimes called the juxtaglomerular complex, which includes the juxtaglomerular cells, also known as granular cells, and the macula densa cells and the distal convoluted tubules. So if the macula densa cells sense an increase in osmolarity, and specifically in chloride because they are designed with chloride receptors, if there's an increase of chloride receptor stimulation, then the macula densa cells are going to stimulate the granular cells of the juxtaglomerular complex, those myogenic cells, to contract and decrease the lumen of the afferent arteriole, because if there's too much solute in the distal convoluted tubule, then there's not enough time for that solute to be reabsorbed in the loop of Henle. And so they're going to cause the cells in the afferent arteriole to constrict, to decrease and slow down the flow of blood through Bowman's capsule, so there's more time to reabsorb the solute. Now, the juxtaglomerular cells also have, they can perform paracrine signaling with adenosine triphosphate, adenosine and nitric oxide to also regulate the constriction or relaxation of those cells. And again, that's referred to as tubuloglomerular feedback. That's a mouthful. So again, looking at the juxtaglomerular apparatus, here we can see the maculodensa cells. They're going to be monitoring chloride ion concentration. Here we have the juxtaglomerular cells. And the renal nerves from the celiac plexus. So those are all going to play a role in the flow of blood through the glomerulus itself. Remember that the juxtaglomerular apparatus is that junction where we have a bunch of cells where the afferent arteriole comes into the glomerulus and the efferent arteriole leaves the glomerulus. And again, these cells are going to be very, very important in regulating the rate of filtrate formation as well as systemic blood pressure. So again, these juxtaglomerular cells, they're modified smooth muscle cells in the wall of the afferent arteriole that are going to respond to help regulate the flow of blood in the glomerulus, and these cells also contain that hormone renin. They also act as mechanoreceptors that contract or relax. That was that contractive response, the myogenic response. And that can also send signals to the cells in the maculodensa. The sodium chloride concentration can vary directly with the flow of blood that's going through the glomerulus. So if there's a higher glomerular filtration rate, that means there will be higher sodium chloride concentration in the distal convoluted tubule, because if the rate is too fast, there's not enough resorption of sodium chloride, and so if there's a lower glomerular filtration rate, that means there will be a lower sodium chloride concentration in the distal convoluted tubule. High osmolarity means that the arterioles are going to contract, decreasing the glomerular filtration rate. Low osmolarity arterioles will relax to increase glomerular filtration rate. And yes, I am having a difficult time speaking. I'll try and get that settled. So the maculodensa, again, cuboidal cells in the wall of the distal convoluted tubule. It monitors the fluid composition, the ion concentration in the distal convoluted tubule, because the maculodensa has chemoreceptors which monitor the solute concentration of the filtrate, especially with sodium chloride. When we look at paracrine mechanisms that control the rate of the blood flow through the glomerulus, we've got three chemicals that are involved. Adenosine triphosphate, which is not just energy, can also act as a paracrine hormone, adenosine and nitric oxide. And you'll notice that when there is increased glomerular filtration rate, which is the first and third of the lines in terms of these chemicals, when the release of adenosine triphosphate, adenosine, is increased, as well as the release of nitric oxide on the third line, You'll notice that the first two cause vasoconstriction and nitric oxide causes vasodilation. However, please keep in mind that adenosine triphosphate and adenosine are stronger than nitric oxide, so they win. There's vasoconstriction. In the condition of decreased glomerular filtration rate, now we have a decrease in adenosine triphosphate and adenosine, as well as a decrease in nitric oxide. However, adenosine triphosphate and adenosine are stronger, and that's why there will be an overall vasodilation. They win. They're stronger than nitric oxide. Everything contributes, but nitric oxide is just not as strong as adenosine triphosphate and adenosine. In terms of endocrine regulation, this takes us back to the renin-angiotensin-aldosterone system, where renin is a hormone produced by the granular cells, which are also the juxtaglomerular cells of the afferent arteriole at the juxtaglomerular apparatus, or some books call it the juxtaglomerular complex. That converts the protein that circulates in the blood that was produced by the liver, called angiotensinogen, into angiotensin 1, which, remember, renin is released due to the juxtaglomerular cells in the presence of nitric oxide. So even though nitric oxide doesn't play a big role in terms of vasoconstriction, vasodilation, it's important in helping cause that release of renin when there is decreased fluid volume. So angiotensin converting enzyme, which is produced in the lungs, will also cause the conversion of angiotensin 1 into angiotensin 2, and it binds to the afferent arterioles and the glomerulus, converting angiotensin 1 into angiotensin 2. Angiotensin 2 is a very fast-acting vasoconstrictor, which helps to reduce glomerular filtration rate as well as reducing renal blood flow. And that's, again, this whole thing is stimulated by a decrease in blood pressure, and it prompts the release of aldosterone, which causes the collecting ducts to reabsorb sodium, which water will follow when sodium is reabsorbed. We know that antidiuretic hormone can help retain and recover water, because when osmolarity increases, if there's too much solute in blood as blood is flowing by the hypothalamus, there are osmoreceptors in the hypothalamus that would detect that increase in osmolarity and cause the hypothalamus to have the posterior pituitary secrete antidiuretic hormone. Antidiuretic hormone will stimulate aquaporins to move into the cell membranes of collecting ducts, and the water will be reabsorbed because it's going to be attracted by the high concentration of solute in the medullary region of the kidney, so that when we have antidiuretic hormone, it's going to cause water resorption and less urine will be formed, because remember, antidiuretic means don't pee. We're basically conserving fluid by increasing the concentration of urine and keeping the water in our bloodstream. So water is going to move out of the collecting ducts and distal convoluted tubules as well into the medullary region of the kidney, where it will be reabsorbed into the bloodstream. There are also other hormones known as endothelins produced by the endothelial cells of renal blood vessels, as well as the cells of the distal convoluted tubules. And these endothelins are released in the presence of angiotensin 2, as well as other chemicals, bradykinins and epinephrine. They're typically not a factor in healthy people. They're released when people are experiencing diabetic kidney disease, and they diminish glomerular filtration rate by damaging the podocytes and by causing powerful vasoconstriction in both the afferent and efferent arterioles. So endothelins will be released not only in diabetic kidney disease, but during and sometimes with some people who have cancer, pulmonary hypertension, arterial hypertension, atherosclerosis, heart failure. Because we have blood vessels throughout our body, these endothelins could potentially be released. Other endocrine regulating hormones include natriuretic hormones, and these are hormones that actually cause the kidneys to excrete sodium, and if they get rid of sodium, water is going to follow. These act as antagonists to aldosterone. Remember, aldosterone causes us to keep sodium and then water follows, so we increase blood volume. Well, natriuretic hormones work in the opposite direction, so they actually inhibit the release of antidiuretic hormone. Atrial natriuretic hormone is a big player in terms of natriuretic hormones because it will be released by the atria in response to their being overstretched. If the atria have too much blood in them, they think there's too much blood volume in the system, and they'll release atrial natriuretic hormone, or atrial natriuretic peptide hormone, sometimes it's referred to, in response to that overstretching. And what that causes is it actually increases glomerular filtration rate by dilating the afferent arterioles and constricting the efferent arterioles, so that there has to be a higher pressure in the glomerulus, causing more fluid to go out of the glomerulus into the capsule, creating more urine. There's also a B-type natriuretic peptide that's produced in the ventricles. It's not as effective as the atrial natriuretic peptide hormone, and it has a longer half-life, so it will last longer, but it will not be as effective. If B-type natriuretic peptide hormone is in the bloodstream, it's usually an indicator of congestive heart failure. Remember that parathyroid hormone is secreted by the parathyroid glands in response to low blood levels of calcium, and they stimulate osteoclasts to break bone down to release hydroxyapatite, and hydroxyapatite is calcium with phosphorus. We don't need excess phosphorus, we need increased calcium, and so the parathyroid hormone will target the proximal convoluted tubule to increase the reabsorption of calcium while blocking the reabsorption of phosphate. We don't want calcium phosphate because that would reduce circulating calcium levels. We just want increased calcium. So it actually blocks the reabsorption of blocks the reabsorption of phosphate while increasing resorption of calcium. And that's how the parathyroid hormone acts on the kidneys. So again, we've got parathyroid hormone being produced and secreted in response to decreased calcium levels. It's going to target the proximal convoluted tubule, where it not only stimulates the resorption of calcium, it also stimulates the hydroxylation of calcidiol to calcitriol, forming the active form of vitamin D. It blocks resorption of phosphate, causing us to lose phosphate in the urine, and by getting rid of phosphate, we have higher circulating levels of calcium in our blood, which is what is required when they fall to levels that are too low. Remember, we need calcium for nerve impulse conduction. We need calcium for the heart to contract, for skeletal muscle to contract. So having a high enough level of calcium in the bloodstream is really important. In terms of what's happening in the distal tubule segments of the nephron, most electrolyte resorption is hormonally controlled in those distal tubule segments. And that ends the urinary system. Yeehaw! I'll see you guys in the next chapter, which is acid-base balance.